All right. So I come from a land far, far away, from the land down under. And Daphne and Ben had asked us to talk about a call to action. So my first call to action is come to Melbourne, Australia. And if you don't like Melbourne, Australia, and you like sports, it's the sports capital of the world. So this captures my reason for being in Melbourne. It's full of sports, from the Australian Open to the MCG to the football fields and so on. If you don't want to come for sports, come for research. All right, so come to Monash and we have a big platform. And what this picture is trying to tell you there is basically that our research focuses on translational research. So we have the scientists, the biologists, along with our translational physicians working all in the same space. So if any of you out there, trainees or even neonatologists, want to dabble into some research and cell therapies, come to us, please. And none of this work which I'm presenting today could be without my cell therapies group. So this is around 10 of us. It's a growing field. It's hopefully going to get bigger with time. But I'm going to represent my group's work on cell therapies and hopefully take you through a journey of what cell therapies is and give you an example or a couple of examples of what translation means, and hopefully the translation moving that promise of stem cells and cell therapies you might have heard of a lot into actual benefits for babies. So hopefully by the end of this talk, you will all start thinking about cell therapies a bit more. All right, so you might have heard of stem cells or cell therapies a lot. So I want to talk about what stem cells are and what cell therapies are to start with. So stem cells, the word actually means that you have stem-like properties, which is two important aspects. One of them being that the ability to regenerate, to divide propensity, and the other one to be able to differentiate into different organ types. Not all cells which we use are stem cells. So cell therapies is actually a better word for that. And there are many sources of stem cells or cell therapies which we have from the body. Some of them are already innate stem cell-like properties or cell therapy properties, which is basically biological agents which have the property to repair, protect, or sometimes regenerate tissue. But in many cases, we can actually induce somatic cells to become stem cell-like. So that's iPSCs, induced pluripotent stem cells. Our group specializes in gestational tissue cell therapies. So this is tissue which is discarded after the birth of the baby, which is basically the placenta, the amniotic membranes, and the cord tissue and blood. So we have been focusing on these important biological agents which we feel have got significant important stem cell or regenerative properties which we can harness to improve babies' conditions. But before I start talking about our work, I want to talk about very important aspect is knowing your cell or stem cells. So just like we've heard from the speakers before, whatever area you work in, it's very important to know what you're dealing with. Same way in cell therapies, it's about knowing the stem cells or cells which we're using. So this is an example I'm going to take you. So the next couple of slides, I'm just going to take you an example of MSCs. You might have heard of MSCs, mesenchymal stromal cells. But there are lots of different types of MSCs, and I'll come to that in a minute. But knowing what cells do is really important. So this is an example of an MSC kind of classification or criteria which people used to ensure that people understand what kind of cells they are using. So the properties which highlight what an MSC can do is their ability to differentiate into different organ types or tissue types like adipocytes, chondrocytes, and so on, and their ability to have surface markers which you can actually replicate in a lab, reproduce in a lab situation. So if you have a cell type and you do not know what it does in an in vitro situation or an in vivo situation, it's very likely you do not know your stem cell or cell very well. So I would say none of us should be involved in any cell therapy or stem cell work or any biological work for that matter unless we know the agent which we are using very well. What do they do inside the body? What do they do outside the body? That's really important. And why is that important? Because when we talk about mechanisms of action, this is a busy slide and I do not expect you to read through that. But if you look at different animal models or preclinical models of health and disease, we have number of conditions which have, for example, inflammation being part of it. And what cells might be doing is kind of decrease or dampen that inflammation. But at the bottom of the slide, what I'm trying to show here is that the MSCs can be of various sources themselves. So just saying that we are using an MSC type to improve this condition or that condition is not enough. We need to know how they are actually working. And what this slide was going to show you, which is I understand it's difficult to see on those screens, is that not all MSCs are equal. So you might say that I'm using MSCs for my cell therapy or stem cell trials. but what type of MSC is it? What is the source of it? And what does it do in those conditions which I talked about in the in vitro and in vivo conditions is really important, because when we start replicating them or actually translating them into human trials, if we do not have good evidence of how they work and how they interact with human tissue, it's very likely that we will not succeed. So knowing your stem cell is my first call to action. Knowing them well and then understanding how they work is really important. So in the next few minutes, I'm going to talk to you about our cell therapy program at Monash. We are one of the world's most active cell therapy trial units in anywhere, and we've completed 3 trials, and we are currently running 3 cell therapy trials. And I've divided them into autologous and allogeneic. Autologous means the baby's own body tissue or stem cells or cell therapy type, and allogeneic means a donor stem cell or cell therapy. What we have worked on is cord blood derived cells and amniotic cells, and I'm going to take you through the journey of those 2 cell types just to give you an example of what translating promise into benefits means. So I'm going to start off with amnion epithelial cells. So the amniotic membrane is the membrane which is covering the placenta, the amnion, the chorion, and the placenta. The amniotic membrane is a rich tissue type which contains cells called amnion epithelial cells, which have the properties of being able to actually differentiate in vitro conditions to different tissue types. So it's like a mesenchymal stem cell, it's like a mesodermal stem cell, where it has that potential of differentiating. What is cool about the amniotic membrane is that it hasn't got much immunogenic properties. So when you give amniotic cells from a donor individual to a baby or an adult or anyone else, it doesn't lead to rejection like a transplant would lead to without matching and so on. So the HLA markers are in very low propensity, and they do not lead to any problems when we give them those cells. Now, it's easy to say this, but you need to prove them in trials first, isn't it? So why did we think about amnion epithelial cells? So like I said, in that Monash Hudson platform I was talking about, where translation happens through a number of preclinical work and a number of lab studies, we showed that these cells in a BPD model, bronchopulmonary dysplasia model, in babies in preclinical environments were of multiple beneficial properties. They were immunomodulatory, they were activating endogenous cells, they were leading to anti-inflammation, they were cytoprotectic, antifibrotic, and so on. So we wanted to translate this work into humans. So what I'm presenting here in a couple of slides is basically 10 years of work, which took us to our first clinical trial. So this was the first in-human administration of allogeneic amnion epithelial cells in premature babies with bronchopulmonary dysplasia. It sounds like a small trial. It was a safety study, just included 6 babies, but it was a giant leap. This was the first time any human had got cells from the amniotic membrane given as a cell therapy. And after this trial, we have got around 15 trials in humans of all kinds, adults, children, and so on. But the first human to get it actually was a preterm baby with significant or severe bronchopulmonary dysplasia. So very proud of this work. This is Rebecca Lim, who actually is now based in Houston, and she works for a big pharma. But basically, we started off with this safety study, which showed that it was safe to give these cells to these babies, and we published their 2-year outcome. So we followed up these babies for 2 years, and we actually following them up longer term as well, but only first 6 babies to get the wheels in motion. The journey is more important than the outcome here. It's about translating that work from the bench to the bedside. And now what we have done is we have taken this to the next level. We have taken it to a dose escalation study. We started off with giving them a low dose in those first 6 babies, and along with one of our PhD students, Liz Baker, we moved it on to a dose escalation study where we gave much higher amounts of those cells over stepped kind of a dose escalation pattern. This study is finished, and we've got a safe dose which we know may be protective for babies who have got high risk of BPD at an early age instead of before they develop that BPD disease itself, so in the first couple of weeks of life. But we're still working in the lab, trying to work on other aspects of the amniotic cells and amniotic membrane. What if we did not give those cells? We just gave the important material which is contained in those cells, the extracellular vesicles, which I'm hoping you would have heard about in other contexts as well. where basically these are the small packets of material in those cells which have the potential to have the same effects but in a cell-free manner, so that the amniotic membrane cells don't need to be given. It is the extracellular vesicles which can be given. So we are doing a lot of preclinical work to move this into trials, but we are probably around 4 or 5 years away before we actually move them into trials. But this is where the amniotic space or epithelial space cell therapy is going into. We've decided that we are not going to move this into the amniotic epithelial cells into an RCT at this stage, but focus on the extracellular vesicles, because we feel the amniotic cell therapy itself has got some glitches, not from a safety point of view, but from a production and off-the-shelf capability point of view for us to use them clinically in a large scale. So that's the journey about amniotic epithelial cells from our lab. Next, I'm going to take you through umbilical cord, blood, and tissue. So the umbilical cord, blood, and tissue, as you can imagine, is the lifeline of the fetus while they are in the womb. But what happens to them afterwards? So the amniotic membrane, similarly, the umbilical cord and tissue has got those stem cells and progenitor cells which sustain life, which regenerate life and protect a lot of us. Especially, you might have heard of babies suffering a brain injury or a significant injury in utero, and many of them actually heal themselves by the time they are maturing. Obviously, the severe ones unlikely to heal themselves, but those cells contained in the umbilical cord and tissue have got significant properties which we can harness. So Lindsay is one of my PhD students who's been looking at the different actions or properties of these umbilical cord and tissue cell types, and we can divide them into 3 main kind of areas which the work has been predominant in the last few years. It's protecting the lungs, protecting the brain, and protecting the heart. And there are a number of trials going on around the world, but I want to take you back to the preclinical side of and tell you the basically journey from our own lab. So Courtney McDonald is my co-lead of our cell therapies group. We always visioned it as a combined work of a scientist and a clinician working together, and that's why we've got our co-leads working together on this. And what we've been working specifically with the cord blood has been the cord blood cells which the cord blood has. So the cord blood, as you might be aware, has got a rich source of hematopoietic stem cells. So these are the most important stem cell variety in cord blood, which basically leads to our bone marrow transplant, which is used in transplant medicine. So the cord blood has got a number of cells, and those cells basically come from the buffy coat. So if you took cord blood and spun it down, you'll get the RBCs at the bottom and the plasma at the top. In between is the buffy coat. And in the buffy coat, we have a number of different cell types. The most important stem cell type is the hematopoietic stem cells. But there are a number of other cells, the mesenchymal stromal cells, the endothelial progenitor cells, the T regulator cells, and so on, which all in combination we have shown in the lab, in vitro, and in vivo models of health and disease to be of really good benefit. And we have been harnessing that in our clinical trials. So this is just an example of the preclinical work in a systematic review from one of my students, where we combined all the preclinical work which is present not just from our lab but from around the world, and focusing on perinatal brain injury acquired in the fetal or neonatal period. And what we have shown in these preclinical trials is that they are beneficial for almost every perinatal brain injury type across various pathological and neurodevelopmental markers. So I don't expect you to read through all the numbers in here, but what I'm trying to convey the message is there is strong preclinical evidence to suggest that umbilical cord blood cell therapies is beneficial for perinatal brain injury. And what we've been doing is trying to translate that promise of the preclinical work into clinical medicine, because very often we see a lot of promise in preclinical medicine, but it does not translate into clinical medicine. This is an example of the cord blood being used for preterm lung injury. So that was perinatal brain injury. This is preterm lung injury. And what I'm trying to show through this slide is that the number of studies which have been done, which I think is still very low. So 23 preclinical studies, mostly in small animal models, some large animal models, and then 6 clinical trials of 100 odd patients in the babies looking at umbilical cord blood therapy for preterm lung injury. Our lab feels that the benefit for the umbilical cord blood cell therapy is more for brain injuries instead of lung injuries. But we obviously know that when we give these cells or any cell therapy to a human, it is holistic. So it's not just going to have an impact on the brain problems, it's going to have an impact on a number of other issues as well. So it's important to keep working on those preclinical trials, understanding them better, as well as working on moving those therapies into the clinic. So the next few slides I'm going to talk about our cord blood work, which is now at an RCT or randomized control level. So, but it started off with the cord safe study, which was basically autologous cord blood derived cells in extremely preterm infants. We wanted to first see if it is actually feasible to even get enough cord blood from these small tiny babies and then give them safely their own product, which has been taken out from their body and processed and kind of enriched to some degree by concentrating them. So this is a busy slide again, but I'll just walk you through it. Cord blood is taken at birth from these extremely preterm infants after cord clamping, whatever the protocol is, and we get that cord blood and concentrate it and get that buffy coat out, get those cells, cryopreserve those cells, and when the baby is ready, generally in a week or two of life, once we have done all the safety testing which is required to be able to give that product, we are giving those cells back. So Lindsay has shown that we have more than 70% feasibility. These charts show that in more than 70% of cases where we attempt cord blood, we get enough cord blood for those babies to be able to extract those cord blood cells to be then able to administer them. And now what we have done is we have shown that it's safe, and that publication is just coming out from the cord safe trial, that it's safe to give these babies those cells back to them in the second week of life. So we've moved on to the cord cell RCT, and there are around 8 North American centers involved in this RCT as well. We have funding to start the trial in Australia, and it's just about recruiting our first patient this week. And in the next few months and years, it will start spreading around to North America, a couple of sites in Asia as well, along with other sites in Australia. And the idea is that given that this is still an early phase trial, we are going to be doing it as a randomized control trial. These are babies whose own cord blood is taken from them, and because we do not know how much injury they're going to have, we have clinical consumer input into them, that we are able to randomize them into a placebo arm and an intervention arm. And the idea is to improve their survival free of major neurodisability at 2 years of age. So watch this space for this RCT, and hopefully I'll have some more of you interested from this conference and other conferences coming to us and wanting to be part of this. We know that although autologous cord blood is a good option for most babies, like I said, 70 to 80% of babies might have that option of autologous cell therapy, but there are a number of babies who will miss out, and these are babies who are born sometimes in difficult situations at home, in a small hospital, or we could not collect enough cord blood. There might be babies who are not born so extremely premature, who still have major brain injury. So we are doing the allo trial, which is an allogeneic cord blood trial, where we are using donor cord blood from publicly stored cord blood bank in Australia. This is the first time anyone is using publicly stored cord blood for a research purpose, and we had to go through a lot of hoops through government to be able to access that, because that was being used for clinical use, but for the first time we are using it for research in babies with severe brain injury. And I'm happy to report that we have recruited 4 babies so far, and all of them have got no safety concerns. Now whether they have efficacy concerns, time will tell, but watch this space where we try to move allogeneic cord blood cell therapy for severe preterm brain injury, which, as you heard from Kimberly just before, when we have severe grade 3, 4 IVH and so on, we do not have any therapy for most of these babies, and many of them succumb, unfortunately. So hopefully moving this space. We are working on cord blood, autologous, allogeneic, but we know that because there is going to be not enough cord blood for many of these babies, we are actually working on expansion as well. So what we are trying to do is actually expand those stem cell or progenitor types in the lab, and then do in vitro and in vivo experiments while we do our cell therapy work, to see if we can have a unlimited supply of these cells which is available for future use. So watch this space for our upcoming work. Not going to be in clinical trials for a while, but just give you an example of the journey of what our cord blood work is like. So in summary, cell therapy shows promise, and hopefully it will start showing benefits. We are hoping in the next few years when we do careful research from preclinical and clinical studies. But I want to still reiterate that point that knowing the stem cell which we're working for is really important. My last call to action is if you are interested in cell therapies, come to our own symposium, which we've been doing in Australia and hopefully coming soon to somewhere in Europe next year and possibly in America in a couple of years' time. Yeah, very happy to connect with any of you interested in cell therapy work. Thank you so much.